Okay, hello everyone, and thanks Molly for inviting me here today. So for a while I've been starting all my talks in a similar way, saying that in my lab we're interested in studying the wonders of evolution, ranging from like speciation and diversification across like adaptation to challenging environments, but that ultimately mutations cause this whole thing and they happen on the molecular level. So what we're trying to do is somehow to integrate all those levels. And so when Molly invited me and she said like, oh, it's like tentatively called like mutation, adaptation, and speciation, I was like, yay, that sounds great, because that's exactly what we're trying to do. And going across these different levels and using modeling, statistics and data analysis, and also increasingly experiments to work on this. But then I saw the schedule and I got super intimidated and increasingly anxious about what I should talk about, because there are so many experts in this room. So yeah, like X numbers of days ago, I then finally decided to give you like really quick snapshots of 2 different projects, like across this continuum, and then focus on one main project, and I'm going to tell you why also. So in this, on the side of mutation, my postdoc Ines, she has been working on studying the distribution of fitness effects and how it varies across environments and along the genome. And so what she's looking at is deep mutational scanning data, where we currently have a new data set of all amino acid changing mutations in the yeast heat shock protein 90. And here you can just see a sketch of the distributions of fitness effects, like the selection coefficients that we estimate, and the proportion we find them in 6 different environments. And so what we're interested in is figuring out in which regions of the protein these fitness effects are environment dependent, and in which way they are environment dependent. For example, whether there's a cost of adaptation where mutations change their sign depending on which environment they are found in. And so this is a collaboration with our colleagues at the University of Massachusetts Medical School. And so, for example, what Ines has done last year was to basically look at this in a slightly more complex way, where she looked at the fitness landscape that is created at a single amino acid position, where you theoretically get to a fitness landscape of all 64 codon mutations that are then kind of connected based on whether they are accessible through single nucleotide steps. And then just kind of intuitively, or like kind of it gives you a fitness landscape that introduces epistasis and additional fitness peaks as soon as you allow for differential effects between synonymous mutations. And in fact, she also saw that a signal that synonymous mutations in HSP90 tended to have stronger effects at high temperatures. Okay, so going to the next level with respect to adaptation, my programmer, Mark, he has been working on simulations to study whether we could eventually predict adaptation of influenza to mutagenic drugs. And so here's a simulation of a genome with like time on the x-axis and differently colored alleles and their frequencies, and the solid line is the population size, and here is just a very small population over time. And if you now give a mutagenic drug theoretically to that population, it should eventually die unless there is evolutionary rescue occurring, which corresponds to adaptation to this mutagenic pressure. And so what we have been doing is to model and simulate these clonal dynamics and identify different mechanisms by which the virus could develop either resistance or tolerance to the drug. And we are now collaborating with a virologist to create candidate mutations that we think should have these mechanisms in order then to test whether they actually have this rescuing identity that we proposed. But I don't know, I still don't know if it was the right decision, but I want to focus today on the speciation part of the work in our lab, and more specifically on hybridization. And so hybridization is interesting to us because firstly, it looks like there's actually a lot of hybridization, and I think that there's more and more evidence that hybridization occurs ubiquitously. But at the same time, it's usually thought to be detrimental. And I think probably most of you know this famous example here from the Xiphophorus fish, where there's an interaction between a tumor gene and a repressor gene that leads to eventual deleterious effects in the F2 generation. And what is known is that there are actually many such genetic incompatibilities across different species. And they are also thought to be important isolating barriers one species have established, but we still don't know how much they really contribute to the process of speciation. And on the other hand, there have been an increasing number of examples where people have proposed that there might be hybridization might actually result in the emergence of new species, like in these butterflies here, where a single gene in hybrids leads to premating isolation to both parents. And this is a case of hybrid speciation. And that's what I will talk about more today. So just to sum up this introduction here, hybridization can result in many different things, and this is particularly interesting for me as a theoretician, and we can formalize this in a way where hybridization can result in extinction of one or both species due to just kind of deleterious load. It can lead to the collapse of species, so-called reverse speciation. It can lead to introgression of alleles, and we've already heard a bit about this today. It might lead to just stable hybrid populations and in the form of clients, for example. It might also lead to hybrid speciation, where we increase the number of species due to hybridization. And it has also been proposed that it can provide the fuel for adaptive radiations, where hybridization might actually promote the radiation in too many species. What we are interested in is whether we can actually predict this outcome somehow and figure out the conditions for either of these, and whether we can find out whether in a specific case hybridization is more likely to result in the loss or in gain of biodiversity. And so the project I'm going to talk about today addresses the question how probable hybrid speciation actually is. And this was led by my postdoc, Alex, who is generally interested in the contribution of genetic incompatibilities and local adaptation in speciation. And I chose to talk about this today because this project was actually inspired by Molly's and Peter's work, and I thought that the probability that while the project is still fresh, I get those two in the same room so I can, thank them for giving the inspiration for this project is quite low. So what we wanted to look at is basically based on this Dobzhansky-Muller model and on genetic incompatibilities that cause negative epistasis. So just to get us all on the same page, we're always talking here about the diploid model, and in the simplest version of the Dobzhansky-Muller model, we have two different loci, which here are small a and small b, and the ancestral version is colored in black. And then during divergence, for example, in allopatry, there might be just new mutations fixing in the population, creating those capital letter alleles here. And then upon reconnection of the population or hybridization, once recombination occurs and those mutations are brought together, or in the codominant case, even already just by forming an F1 generation, we might get these incompatibilities to appear. So in terms of a fitness scheme here, this would be our ancestral type, and we assume that the derived alleles are on their own at least have the same fitness or a higher fitness, but if they are brought together into the same genome, they are deleterious, which builds this DMI or Dobzhansky-Muller incompatibility. And for all the results that I'm going to show today, we assume that there's a very small selective advantage, direct selective advantage of the derived alleles of the order, kind of 2 orders of magnitude lower than the actual epistatic effect. So just for you to know. So what Molly and Peter proposed was that if we have such incompatibilities present in the genome and hybridization occurred, we can get something that they called reciprocal sorting of those incompatibilities, where either of these incompatibilities will be resolved eventually because it causes deleterious effects. But if certain genotypes fix, they might result in isolation to both parental species. So these, in this case of two incompatibilities or 4 loci, you can get 2 different what we call hybrid speciation genotypes, where one incompatibility is fixed for one parental species, for one derived allele from one species, and the other one for the other. So if you backcross this to either of the 2 ancestral populations, you will get the expression of one of the incompatibilities respectively. And so that way, this new population, which is now fixed for this genotype here, will be isolated from both parental species. So what we set out to ask is, generally in like a very minimal model, how probable is this reciprocal sorting of DMIs, which is also partly what Molly has been addressing, but also what is the role of the linkage architecture, which is the arrangement of the different incompatibility loci in this process. So the simplest model to actually write this, to do this, has 4 loci because we need 2 incompatibilities. So here we have like these are the 2 starting basically the haplotypes of those 2 starting populations. And here's just one example where here we will always assume that the capital A1 and B1 alleles are incompatible, and the capital A2 and B2 alleles also. And so if you write down this model, you notice that there are actually 6 different linkage architectures that can lead to this, that can happen here, which depend on the organization of the A and B loci, and also whether the incompatibilities, so if they are on the same chromosome, then you have these 6 different organizations. And of course, if each incompatibility is on a separate chromosome, basically that's kind of what this all converges to with increasing recombination rate. And so the first thing we found was kind of boring and very consistent with what everyone had been proposing before, namely that hybrid speciation or reciprocal sorting is very rare. So here on the x-axis, I'm showing you the initial proportion of the genomes of the 2 parental species, and on the y-axis, I'm showing you the hybrid speciation probability. And this shows you that for quite like intermediate, like strong epistasis and free recombination in a reasonably large population, hybrid speciation is really quite rare, and it only can ever occur if the initial proportions are very balanced. But then Alex, he started to look a little more closely into those different linkage architectures, and at different recombination rates if incompatibilities occur on the same chromosome. And so here, for example, he assumed that it's still quite rare, although it becomes a little more likely if the population size becomes larger. But then once he looked at different linkage architectures, he found cases in which suddenly reciprocal sorting became almost deterministic. For example, in this one here. And this can happen always whenever recombination is at intermediate rates corresponding to about 1 recombination event per generation. And so I think it gave Alex quite a lot of sleepless nights because he really wanted to explain what was going on. He checked his code and for potential bugs. But like eventually he figured out what is really going on. And but first to show you, he found that this is not just a random occurrence, but in 2 out of the 6 scenarios, we saw this really kind of very likely occurrence of hybrid speciation. And now to kind of solve what's going on here, it's interesting to look at the F2 breakdown. So after the first recombination event appeared, and which types of haplotypes are created in the population. So if recombination happens between, at this point here, we get those 2 haplotypes. And as you can already see, this one has a higher fitness, leading to a higher marginal fitness of this red allele here. And this one has an incompatibility, so it will be strongly selected against. if recombination happens at the second point here, then you can get the 2 hybrid speciation haplotypes. So these will be equally fit in the population. And if recombination happens at the third position here, again, you get 1 incompatible type and another single allele, like a blue allele, that is fitter than this other haplotype. And so what is now going on is that these 4 here, they are the ones that have no incompatibilities. And so, and also, and when either of these again recombine, they will always be fit because this is the hybrid speciation haplotype that has no incompatibilities left. And so that enriches the population immediately, no matter where recombination occurs in these different haplotypes, then eventually leading to fixation of the hybrid haplotype because it has a slight selective advantage. And just to compare this with the other scenario, in this scenario here, for example, for the other linkage architecture, you get 4 incompatible haplotypes. And while you still get those red and blue types, whenever they recombine, they are selected against because of the incompatibility. And so either only one of those can ever fix in the population. Okay, so what we saw was that this marginal fitness advantage of the epistasis-free haplotypes can lead almost certainly to hybrid speciation in large populations. And then the second thing that we looked at was this initial proportion of the different parental populations. And intuitively, you would always, since all our selection coefficients are symmetric, one would expect that also symmetry should be the best case scenario for getting reciprocal sorting. But then once we started looking at those initial proportions and the hybrid speciation probabilities, we saw that this is not always the case. So here's again our one of the linkage architectures, and here's now the initial proportion of the different parental populations and the recombination rate now on the y-axis. And so you can see this rather low hybrid speciation probability, which increases with recombination, and it's centered around symmetric contact of the 2 different populations. And then the one that I showed you before, where we have a very high recombination probability. Now you see that, so this is now on a log scale, so it looks a bit different than before. We still get this really high hybrid speciation probability, but only for symmetric cases. Now, finally, if we look at the third type of linkage architecture, we get an asymmetry in the picture. And again, this can be explained by the F2 breakdown here, where now if we build the possible recombinants, the fit epistasis-free haplotypes are both of the red parental, both of those that have the red allele. And so hybrid speciation in this case is maximized by balancing against these red types. So if you start with more of the blue ones, you're much more likely to ever recombine into the hybrid speciation haplotypes. And that leads to this asymmetry as an ideal starting point for hybrid speciation. So this already leads me to the conclusion. So as everyone has been, I think, not denying so far, hybrid speciation is usually rare, but it seems that for specific linkage architectures, it can be almost unavoidable and potentially repeatable. And of course, so what I've shown you is that you need very large population sizes for this. So probably like microbial populations, for example, like yeast or so, might be much more likely to show this repeatable pattern than like a very small populations of other organisms. And we also saw that symmetric contact is not always the best scenario to observe hybrid speciation. And in all of these things, this can be explained by a critical role of this initial F2 breakdown of the hybrid population. And yeah, this is still kind of fresh. So if you want to read more about this, you can look it up here. And there's also a slightly kind of condensed version, like nice kind of version online for like a more general audience. So with this, thanks again to Molly and to all of you for staying awake despite jet lag and everything. And I'm very grateful for my great team in Portugal, and especially for Alex's work on the project I was mainly talking about, and to my collaborators who are always up for discussions. Thanks.